Okay, moving on. Let's do mast cells before we do natural killer cells, because natural killer cells are a little bit involved. So let's do mast cells because they are fairly easy. Excuse me. So mast cells generally sit in the lining of your epithelial cells. So going back to our lung example, you might have, some epithelial cells. And then what you might also have is a mast cell in between, which we'll draw in blue. In here, maybe. And mast cells are very much, well, they're not very much like neutrophils, but they are like neutrophils in the sense that they also have pre-formed vesicles within them. And these vesicles contain factors like histamines, if you've heard of an antihistamine for hay fever. This is what it's acting against. Something called a leukotriene. Can never spell that, is it? I-E-N-E. Anyway, I hope it's that. And these cells are able to degranulate themselves. If we kind of zoom in on a mast cell, so for example, okay, that's the nucleus, just for fun. What they have specifically on the outside of them, which is quite interesting for an allergy perspective, is they have antibodies on the outside of their surface. These antibodies are specifically called IgE. There's lots of IG insert letter. So IgE. And we'll make sure that we've got the granules in there as well. So having their surface these IgE molecules. And what we find is that actually those IgE molecules can be cross-linked by something like a pollen, say like in the case of an allergic reaction. Not drawn to scale. What will happen is you'll get cross-linking of these IgE molecules that are specific for pollens and things. And that will cause the mast cells to degranulate. Now, obviously in the context of allergy, that's not good if you're allergic to grass and birch pollen and such. And in fact, if you're allergic to animals as well, these IgE molecules could be specific for allergens of a variety of different sources. So allergen specific. It's thought that they've actually evolved. They've obviously not evolved for allergic purposes. That would be quite counterintuitive. But they've been thought to have evolved to also degranulate to things like pathogens and worms. Now, in developed world, we don't really have that kind of problem with larger pathogens and worm infections due to sanitation, water, clean water, that kind of thing, clean food. But obviously we never used to, thinking thousands of years ago. And it's thought that this response is actually directly evolved to counter those types of infections, but actually we don't see that often. But they're still important. These histamines and leukotrienes, what they can do is one of the biggest things that they do is they cause vasodilation, it's called. So vasodilation is when you have, say you have a blood vessel, right? A blood vessel is actually lined with these types of cells called endothelium cells, right? So lots of theliums. Okay, so this is your blood vessel. This is a normal capillary that's going through, say, your skin, for example, blood. What can happen is things like histamines, leukotrienes, and vasodilators cause your vessels to become a bit leakier. They don't like punch holes in them or anything. They're just, the gaps between the cells can increase slightly. And that's called vasodilation. And let's see, the blood kind of flows through. Now, why is that important? Why is vasodilation important? Because if you dilate your blood vessels, your blood pressure drops within that, because you've now got a larger, wider vessel for blood to go through, so the pressure is going to be less. And it can also allow for other immune cells to come out of the circulation, out of your bloodstream, white blood cells, to enter into the tissue that's possibly being infected, which is really important. Okay? So you get migration of immune cells. And that's one of the key factors that goes on. Okay, so the final thing on the cells is we're going to talk about NK cells. And I'm going to do my best to explain something fairly complex. Wish me luck. Natural killer cells. Okay, I've just drawn a big line all the way through that. Natural killer cells. So effectively, the immune system is only really able to recognize something that is not yours, not human, not your own. It does this indiscriminately, really. There are methods for adaptive immune responses where you end up with a very specific response. But what the cells are actually doing, and what the immune system is actually doing, is just saying, that's not me, or that's not human, that shouldn't be here, let's get rid of it. Okay? And this is why when you have, so the immune system doesn't really care if it's a bacteria, if it's a virus, or even if it's a donor piece of tissue. So if you've heard of tissue rejection, if someone has a transplant, like an organ transplant, you may have heard of the term rejection. And what that is it's a process of the recipient's immune system slowly getting rid of this organ, rejecting this organ, because the organ's not from that person, and the immune system is trained very early on to not attack itself, but to basically attack everything else. So that's why you need tissue matching, blood matching when you do organ transplants, because the immune system knows that it's not itself that it's seeing. It doesn't know what it is, but it knows it's not itself. And natural killer cells do this quite well. Something we haven't really touched on is every cell in the body. So epithelial cells, even the immune cells, but generally liver cells, lung cells, brain cells, this type of thing. They have on their surface something called an MHC class 1. So let's say this is a human cell. And we have inside and outside. And what we have on this is, we'll draw this in yellow. Hi folks, sorry to interrupt, but I wanted to thank you for making it this far through the video, and to let you know that I really appreciate your engagement and I hope you're enjoying the content. Everything that you're watching here was live streamed over on my Twitch channel, twitch.tv/4085. I stream 3 times a week, Monday, Wednesday and Thursday at 9 PM UK time. And if you want to get involved in any scientific discussions, ask me any questions, or just watch a bit of Xbox gameplay, you can head over there to do so. This YouTube channel is dedicated to the science content. I have a second channel where I post highlights of gaming sessions and other miscellaneous content. The link to that channel is in the video description. Thanks again for your time. Now back to the video. I should be an engineer. Yes, we have. It's like you can think of it as like a cup, or a chalice, or even a plinth, or a little hand. So, just for the immunologists out there, you'll know what this little square is about. But so this is called MHC1. It stands for Major Histocompatibility Complex 1. Don't worry about the name. It's a bit long-winded. Now, these are on the surface of every nucleated cell. And what they do is they do something called antigen presentation. And what they're doing, they're constantly doing this, right? So they hold a little piece of another protein. And they think of them like a window, okay? So when you're on the outside of a cell, all the proteins and everything that's going on inside the cell at one point or another will be presented on many of these MHC class 1 molecules. And it's like the cell holding up a sign saying, this is what's going on inside my membrane. Okay? And the beauty of this is that when something goes wrong within a cell, something goes wrong within the MHC class 1 presentation, and it changes. The MHC class 1 presentation changes, and you can view that outside of the cell. And natural killer cells are a cell of the immune system that have evolved to recognize this MHC class 1. And if the cell has a proper intact MHC class 1, then the natural killer cell doesn't say, but the natural killer cell sees it and moves on. And if it comes across a cell that has a very abnormal MHC class 1, i.e. representative of something weird going on inside the cell, possibly a bacterial infection, possibly a cancer cell would do this. But what it will do is it will then target that cell for death in quite an elegant fashion. So this is an MHC class 1 molecule. What I will draw is a couple of examples of an infected cell versus a non-infected cell. So we've got a membrane. This is going to be this cell is going to, so let's say out, in, out, in. This cell is going to be infected. So we've got virus infection, for example. So there are viruses inside this cell. Not good. Bad. And this one's fine. So what will happen is that on the surface of the cell, this one will be presenting a normal MHC molecule with normal protein. Now, one of the results so what happens when a cell infects a virus is one of the things that generally happens when a cell gets kind of perturbed or out of whack, or something goes wrong within the cell. What happens is that actually you don't get any MHC being produced, so you actually get none. So we have MHC, and there's also an activating there's an activating ligand as well, which we'll just draw in. So just for this is an activator. So there is another protein that exists on the surface of normal cells as well. Activating ligand. Won't go into what they're called. So the difference between these two is that one has MHC in this activating ligand, and the other one doesn't have any MHC because it's infected with the virus, but it does have the activating ligand. And what happens is that your natural killer cell, which we'll draw in green, shall we? So this is our surface of our NK cell. And this is the opposite way around. So this is the out, and this is the in. So we're looking at the NK cell coming in from above. The NK cell is looking for this. Okay. The NK cell will have, draw in yellow again. Let's draw it in actually in gold. So this will be the activating receptor. And importantly, it has an inhibitory receptor as well, which will bind perfectly on the MHC1. And when it receives the two signals, so in theory it receives an activating receptor and an inhibitory receptor, the result is no activation. However, in a virally infected cell, it's got its activating receptor and what you get, if it doesn't have the inhibitory signal, you know what it's going to do, it's going to activate. Okay. Now, what it does, when it activates, is it actually releases factors from one cell to the other upon activation. And what that does, in quite a macabre kind of way, is effectively the NK cell, this will trigger something called apoptosis. It will trigger apoptosis in the infected cell. And apoptosis is another word for programmed. That's 2M. Cell death. Effectively, the NK cell tells the infected cell to kill itself. And it does. And this is one of the major ways in which the innate immune system can deal with viruses and infections. And it does this all the time. So sometimes you'll be exposed to a pathogen and you won't get sick from it, and your body will effectively clear it up without you even noticing. That's your innate immune system going to town through all of these different methods. So that's going to conclude the overview of the cellular response to the innate immune system. I hope you've managed to follow along with my horrible handwriting on this pad. I'm still not used to this pad. I will endeavour to be better. And in fact, that's going to actually conclude that part of the stream, I think. So what we've done, actually we've done quite a lot. We've gone through trying to find the beginning here, right? So yeah, right at the beginning, we started with innate immunity. We were looking at barriers, complement, and cellular responses. Barriers being skin, GI and respiratory tract, and the like. Anything that kind of touches the outside world. Did a little bit on the complement system. Complement system is a series of proteins that form themselves into a cascade that ends up forming pores in the membranes of bacteria and can kill a bacteria that way by everything kind of leaking out of it. And then you have the cellular responses of the innate immune system, far more involved. You've got loads of different cells: epithelial cells, neutrophils, macrophages, mast cells, and natural killer cells, just to name a few. So that was effectively the overview of the innate immune system. Next week, I'm going to be doing the adaptive immune system, which is even more complicated. And I hope that, you can tune in then and we can kind of resolve this in a nice kind of tight bundle, which will be really, really cool if we can do that. And I may have better drawing skills by then. Who knows? Probably not, but whatever.